Okay, we're going to take a look at experimental design today. We've already reviewed what problem and hypotheses are, so let's take a look. In a controlled experiment, again, you start with the problem, which is a question. Does Elodea need light to make food? Remember, a problem is always a question. The hypothesis is going to try to answer that question. So what would be a valid prediction for does Elodea need light to make food? Take a minute and try to guess. An easy way to make a hypothesis is to think of it as an if-then statement. If Elodea is given light, then it will make food. A hypothesis is a prediction. It's never a question. A controlled experiment. Take a look at the picture in front of you and make some observations. Well, one thing that you should notice right away is that there are two different groups or two different setups here. They are similar with the exception of one thing. What is the one thing that's different? One setup is with light and one setup is without light. You'll notice that we call the light the independent variable because it only exists in one of the two groups. The other group, there is no independent variable, so there's without light. Now, there are other things involved, and we call some of these constants or controls. What are some things that are controlled in this experiment or that are kept constant between the two groups? The water and the green multicellular water plant. So to review, in a controlled experiment, there are always two groups. We call them the control group and the experimental group. Within these two groups, we keep constant factors that are kept the same between these groups, or we control those factors so that they are kept the same. In the control group, there is no independent variable. As opposed to the experimental group, there is the independent variable that exists. The constants or controls in the experiment. Experimental group and control group have the same setup except for the independent variable. This ensures that it is a fair test of the effect of the independent variable. Some examples of constants or controls from this particular experiment were maybe the same number of plants used, the same temperature of the water, the same amount of water. The idea is that everything is kept the same except for the factor that you're testing the effect of, which is the independent variable. In addition to keeping things constant, in science we also need to be able to replicate the experiment to prove that it is accurate or valid. One of the ways we do this is by having a large sample size. A sample size is the number of specimens used in the experiment. One plant for each group would be way too small. Something could easily happen to one plant that could kill it and give us inaccurate data. So instead, we use multiple plants, something like 50 plants or 100 plants. In an experiment, multiple copies of the same experiment or repeating the same exact experiment is what gives us the validity or accuracy of the experiment. The next part to talk about is the dependent variable. The dependent variable is the responding variable or the outcome of the experiment. So what happens after you're done with your experiment? What is the data that you're collecting? Oftentimes when we collect data, we'll collect it in a table. You can see here a sample table with independent variable in one column and dependent variable in another. From the experiment you saw in the beginning, it would be amount of light as the independent variable, so light versus no light. So our experimental group and our control group are listed. The dependent variable in this experiment was whether or not the plant was making food. One of those ways of measuring that is whether or not the plant was making oxygen, so we could count the number of bubbles, 5 being the maximum amount of bubbling, 0 being none, and we'll see that with light, it gave us a 4 for the amount of oxygen bubbles, and with no light, we got a 0. The dependent variable, again, is a measurable outcome. So let's review with some questions. How many independent variables can you test? Only one. How many groups are in a controlled experiment? Two, the control group and the experimental group. And what are controls or constant factors? They are factors you keep the same in the experimental group and control group. So it's everything except for the independent variable.
What is a computer system? It sounds like an easy question. You're probably looking at one right now. But